Hello everyone, so let's begin the next video on mineral nutrition, that is deficiency symptoms of the essential elements. Now we have already studied about the essential elements, isn't it? Total we have got 17 essential elements, but as per the recent studies 3 more have been added to the group, that is cobalt, then silicon and vanadium. Okay, so you besides the 17 plus 3, that is 20 essential elements are there. Now of these essential elements, actually we know that they are very much essential for the growth of plant, isn't it? The growth and development, physiological activities, etc. So they are required in some basic quantities, amount in the plant, isn't it? Some are required more, some are required less. Sometimes what happens, if the necessary amount is not received by the plant for their growth and development, then what happens, certain problems occur. That particular problem is known as deficiency symptoms. Okay, deficiency of externally visible pathological conditions, actually, which are produced due to the absence or deficiency of some essential nutritive substances. It is also known as hunger sign, because the food is needed, isn't it? The plant is hungry for the food, for the nutrient, and it is not receiving. That's why it is also known as the hunger signs. Now, while we are studying about the deficiency symptoms, we come across the term critical concentration. Critical concentration means the minimum amount of concentration of the essential nutrients which is required for the growth and development. Now, you see in this diagram, suppose this is the particular line indicating the critical concentration of a plant. Okay, this particular amount is needed by the plant for its growth and development. Suppose the amount decreases, then what happens? Deficiency occurs. And if that particular amount exceeds, which is to be present, more than that is present, then it is known as toxicity. Okay, so both are harmful, but if deficiency is there, then we can add the nutrients and just equalize the critical concentration. But if toxicity occurs, it becomes difficult for the plants to survive. Okay, so these are the basic things while we are studying the proper amount of minerals required by the plant. So critical concentration, the minimum requirement; deficiency, lack of the nutrients; toxicity, excess utilization of the nutrients. Okay, now there are several diseases of plant. The first disease is chlorosis. Chlorosis means when there is lack of chlorophyll formation, the plants become yellow in color. Okay, the leaves, and this is due to nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc and molybdenum. Then stunted growth. Stunted growth means their growth is not as usual. Okay, and the elements which are responsible for this are sodium, potassium, calcium, sulfur, zinc, boron, molybdenum and chlorine. Then next is the purple coloration of shoot axis and leaves. That means due to deficiency of nitrogen, phosphorus, magnesium, sulfur and molybdenum, some particular parts of leaves and the shoot apex become purple color. The next is necrosis. Necrosis means it is localized death of tissue. That means in the leaves some particular patches dies, and that is called necrosis, and that is due to calcium, magnesium, copper and potassium deficiency. Then next is a premature fall of leaves and buds. Now we know that when the leaves and buds become mature, only then they die or fall off, isn't it? But here what happens, before they mature, the fall takes off. That is due to deficiency of phosphorus, magnesium and copper. Then some particular substances like nitrogen, potassium, sulfur and molybdenum, these are responsible for inhibition of cell division. Then wrinkling of cereal grains, that is caused by nitrogen, sulfur, molybdenum. Dormancy of lateral buds, that means the lateral buds are inhibited. If lateral buds are not developed, dormancy occurs, then what happens? The girth of the plant stops. It will only grow in one particular direction towards the top portion, isn't it? And that is also responsible due to nitrogen, sulfur and molybdenum. Late flowering, nitrogen, sulfur, molybdenum. Then next is dieback. Dieback means what happened? The apex tissue of the apical portion dies, shoot tip, leaf tips, okay? Then what happened? Gradually that portion continues towards the lower part. That's why that is called dieback disease, and that is due to potassium and copper deficiency. Wilting, that is loss of turgor. Water loss, okay, that is due to chlorine. Death of root and shoot tips caused by boron deficiency. Bushy habit of shoots, that is due to potassium deficiency. Scorched leaf tips, potassium deficiency. Interveinal fluorosis, iron deficiency. And victal disease of leaves due to molybdenum deficiency. So these are the all total 16 diseases which we often observe in case of the plants. Okay. Now, after we have come across all these things, now let us go and study how the minerals are absorbing the nutrients. Actually, earlier also while studying the transport in plants, we have studied how the water is being absorbed, isn't it? So, the same mechanism also take place in case of mineral absorption, because water and minerals are absorbed or transported to the upper part from the roots through the xylem tissue, isn't it? That means only it is unidirectional flow. And here the minerals which are present in the soil, that means soil is the reservoir of the minerals. So that passes into the roots, and from the roots to the xylem tissue and towards the different parts of the plant. Okay. Here again, okay, next, if we see that soil is the reservoir of all minerals, isn't it? Minerals are present in the form of ions in the soil. So here the concentration is more. But in roots what happened? The concentration actually of minerals are much more as compared to the soil. Okay. So that's why when the minerals enter into the roots, then what happened? It uses ATP and it moves against the concentration gradient. That's why this particular transport is called the active transport. Okay, that means absorption of the ions into the inner space takes place through active absorption. But if these ions move into the outer space from the cell, then that is through passive absorption. Okay. Now, the mineral absorption, it actually takes place by two phases. One is the initial phase and other is the metabolic phase. In initial phase, the rapid uptake of ions take place, okay, into outer or free space of the cell. That means from the cell towards the outer portion it takes place. And here actually free space means the intercellular spaces and the cell wall. And in metabolic phase, the ions passes into the inner spaces, that is cytoplasm and vacuoles. Okay, so these are the basic two phases for mineral absorption. Now, various theories or examples have been provided for mineral absorption, that is translocation of solutes. Now we know that minerals move from the soil to the epiblema of cells, then to the tracheary elements, that is the xylem tissues. Okay. Then with water actually they move upward. Okay, the same process. Here xylem sap contains the mineral salts which are taken upward. Then salt uptake increases with the increase of the rate of transpiration. Okay, these are actually similar to that of the transport on plants, whatever we have studied earlier. Then ringing and girdling experiment also proves that, help us to study the movement of minerals. Okay. Then another technique is there, the tracer technique. Actually, the barks, they contain more minerals than the wood. Okay, but actually what happened, the phloem is not the pathway of minerals, but we know that minerals pass only through the xylem tissues. So Stout and Hoagland, they studied or performed an experiment in which they inserted a paraffin paper, okay, between the xylem and phloem tissue, and it was and radioactive potassium and phosphorus was added to the rooting medium, okay, with different concentrations were then observed. Later what happened, when they studied the paper, the stripped area, they observed that little mineral concentration occurred in the bark portion and much more concentration was present in the xylem. This was quite expected part, isn't it? Because we know that xylem is the main way of mineral transportation, so definitely xylem tissue will have much more concentration of minerals. But initially we have observed that bark tissue shows more mineral concentration. Now what is it? Why it is so? This is so because the minerals in the bark and above strip, that is due to local absorption of minerals from xylem by the living tissues. That means the bark cells are living cells, so they will just absorb the minerals towards them, and so their concentration increases as compared to the xylem cells. Okay, so these are the all basic things that are important parts that are related to the translocation of solutes. So hope this video is clear to you. Go through it, go through the notes also. If you find any problem, then please let me know.